Malignant myxoid mesothelioma, is a rare and aggressive subtype of mesothelioma that carries a very poor prognosis. Standard treatment approaches including surgery, chemotherapy, and radiation have shown limited effectiveness against MMM. Due to its rarity, there have not been any large randomized clinical trials specifically focusing on MMM. However, immunotherapy has emerged as a promising new therapeutic approach for other types of mesothelioma as well as other cancers. Several immunotherapy clinical trials are actively recruiting patients with MMM or mesothelioma subtypes to test the safety and efficacy of various immunotherapies either alone or in combination with other treatments. One of the earliest clinical trials exploring immunotherapy for MMM is a phase I/II trial testing the PD-1 inhibitor pembrolizumab in patients with advanced mesothelioma, NCT02488759. The trial enrolled patients with epithelioid or non-epithelioid subtypes, including those with MMM, who had progressed on prior chemotherapy. Preliminary results showed that pembrolizumab had an acceptable safety profile and promising anti-tumor activity in a subset of patients with mesothelioma, including partial responses seen in some MMM patients. Based on these results, further studies are warranted to validate pembrolizumab's efficacy against MMM. In addition to checkpoint inhibitors targeting PD-1/PD-L1, other immunotherapy clinical trials are actively recruiting MMM patients. The Mesothelioma phase I/II trial, NCT04101777, is evaluating the safety and efficacy of a trispecific killer engager, trastuzumab, immunotherapy in patients with advanced mesothelioma, including MMM. Trastuzumab works by bridging NK cells and T cells to tumor cells expressing its targets, CD33 and CD123. Early safety results from this ongoing study were favorable. Another ongoing trial, MyPayVac1, NCT04725835, is evaluating personalized peptide vaccination with or without tremelimumab in mesothelioma patients, with MMM eligible for enrollment. This personalized immunotherapy approach involves identifying patient-specific tumor mutations and generating personalized vaccines to elicit an anti-tumor T-cell response. Tremelimumab is a CTLA-4 inhibitor included to enhance the immune response. Additionally, combination immunotherapies are also being explored in clinical trials as a way to potentially improve responses in MMM and other difficult-to-treat cancers. For example, Keynote 792, NCT04199236, is an ongoing phase I/II trial combining the PD-1 inhibitor pembrolizumab with the TLR9 agonist motelimod in advanced mesothelioma, including MMM. The rationale is that the TLR9 agonist may help overcome resistance to checkpoint inhibitors by enhancing antigen presentation and priming anti-tumor T-cell responses. Finally, two major NCI-designated cancer centers, the University of Texas MD Anderson Cancer Center and New York University Langone Health, have ongoing MMM biomarker studies investigating potential immunotherapy targets in MMM tumors and immune profiles correlated with responses, NCT03993366, NCT04828558. This could help identify subsets of MMM patients most likely to benefit from immunotherapies in future clinical trials. While still limited due to the rarity of MMM, existing and ongoing clinical trials provide early evidence and rationale for exploring immunotherapy approaches in MMM. Checkpoint inhibitors, cellular therapies, vaccines, and combination strategies are active areas of investigation. Further clinical research and immunotherapy development hold promise to potentially change the treatment landscape and improve outcomes for patients with this currently difficult-to-treat cancer.